So this talk is essentially it's an attempt at a synthesis of a couple of projects I've been working with in my PhD, and essentially it's a question of deep animal phylogeny, and invariably questions about deep animal phylogeny invoke the Cambrian Explosion in some context, and the kind of the popular view is this radiation of animals in terms of body fossils is also has parallel diversification in terms of disparity, in terms of ecosystems, but quite interestingly, the record of things like the exceptional fossil biota is predated by a skeletonized biota of quite enigmatic fossils, the so-called small Shelly fossils, which have operated as kind of a taxonomic kind of wastebasket, essentially. And specifically, I want to talk about the group called Tomodids. The small Shellies encompasses a wide range of tubes and other things. So generally, the things I've been working on are these sclerotome animals derived from acid-processed carbonate rocks, and we have this incredible variety of phosphatized skeletal elements of uncertain affinity. So what are these things, really, is the question. Do they have any relation to the phyla we all love and know today? Or are they simply kind of curiosities? And obviously they don't resemble, outwardly at least, at the most basic level, animals that we can recognize. So the kind of approach that I've been taking is to kind of look at a detailed character-based approach, looking at things like development and histology, to try and determine some of the homologous character suites. And just to put this into context, the broad consensus is a lot of these things might be Lophotrochozoans, and this is kind of the current picture of what we know about Lophotrochozoan phylogeny, and it's not a pretty picture. Essentially, yeah, the topology of the tree, even from these very extensive phylogenomic studies, is anything but decided. So this is really one of the open questions in biology today: is are Lophophorites a clade or not? So this is somewhere where actually, as morphologists and paleontologists, we can possibly add something to the argument. So molecular clock studies, of course, need calibration points. So this is kind of the common usage of fossils by molecular biologists. But we can also use kind of morphological data to kind of polarize how body plans are assembled at this kind of critical juncture of evolution in the Cambrian. So the conceptual model that the Tomodids have been kind of slotted into, this is a diagram from Skofstad et al., and it's modified by Duncan Murdoch and colleagues, and it's kind of an attempt of taxonomists to kind of place a lot of these taxa into some kind of context, as they had some kind of similarities to lingulid brachiopods or inconel forms, or even pharaonids. So this was kind of the hypothetical tree, and it's not without its problems, as Murdoch et al. Pointed out, that the character support for the schema it's not particularly strong, and that some of the putative homologies don't stand up really well under scrutiny, and they produced a kind of an attempt at a cladogram, and again, this is an unrooted network diagram. It's not a pretty picture. So is there actually any hope? Can we really place these things? And by going back and just kind of looking at the data, just doing hard morphology, I think, yes, we can possibly get a signal if we look hard enough. So I set up this Morphobank project essentially to provide some kind of collaborative platform to build a data matrix, and essentially using broad taxonomic sampling of Tomodids and early Cambrian stem brachiopods using micro-CT and synchrotron tomography. So I'm not going to talk too much about the techniques, but essentially walk you through some of the character suites that we've been working on to try and pull some signal out. So this is the Tomodid micrina, which is a small cap-shaped fossil with, we think, a bivalve sclerotome, and it's covered in these little pores. And these have been kind of, the actual interpretation of these structures has been slightly controversial. So the initial interpretation is that these pore structures are actually, as Tim talked a lot about, Keedy. I'm sticking with the traditional classification as CD, but that's an argument for another day. But essentially this character complex composed of these phosphatic columnar structures and tubes containing conceivably chitinous CD that penetrate through the shell. And this was a character that was essentially flagged as unreliable, but I think by using some of our own micro-CT data, we can very well support this, because when we look at the actual interaction of the columns and the sedal tubes, so this is just a rendering of the tube wall initially, and the argument that these weren't CD was that they were bifurcating, but occasionally you have intersections between the columns and the CD. And this extra structure here we see in white, this is in fact an exceptionally preserved CD, and possibly the cedoblast cell at the base of the sedal tube. So essentially we have the CD in situ in straight tubes, and occasionally the intersections between these two structures may look like they're bifurcating, but if we go back and look at these things with an SEM, they are undeniably, they are CD in situ. So we've rendered a couple of these here in Blender. And yeah, so this is kind of a consistent pattern. It's also quite interesting that you have this kind of micro exceptional preservation within a phosphatic shell of an originally organic structure that pops out in the CT scan. So these are phosphatized CD, which is quite interesting. So we've kind of resurrected this character. And the reason that it's interesting is because this character system actually seems to have quite a wide distribution amongst other tomodids. So we think that, for example, Tanulinids also have these sedal tubes and columns. Sonogynia has columns but no sedal tubes. And the stem group Brachiopod, so much more, actually, yeah, it resembles a brachipod. Mycvitzia also has this pore and column system, especially in the larval shell areas where essentially we have a tomodid-like shell structure in the larval shell. So that's actually starting to tie tomodids to something that's much more brachipod-like. So that, yes, there's data here, and moving from Micrina to an intermediate form Cedatella to Mycvitzia, that there's possibly kind of a narrative here. And if we follow up looking at these pore and CD complexes, this is Helimedusa, a Zhang exceptionally preserved brachiopod. And what we actually find is that this contains both these shell-penetrating CD and also these inward-pointing cone structures, and these are something that also turns up in Mycvitzia. And this, I think, this is up to now been an kind of a tapomorphy of Mycvitzia, but now it's turned up in Helimedusa. So then we can, if these are sister taxa, we can or even the same thing, we can start telling something about, well, yeah, the soft tissue anatomy of these organisms. So this data is being fed into a matrix in Morphobank, and if and when this gets published, this will go live, so it's open to scrutiny, and anyone can download the data sets, use it again. I've run my own phylogeny based on this data set, and also produced a splits tree network, so those were just the parameters, just to provide a direct comparison with the Murdoch et al. Data set. So with kind of slightly more taxon sampling. And essentially what we find is that you can broadly cluster the things that pop out into the brachiopod groups that we all know and love. And because it's an unrooted network diagram, there's no assumption of where to root this tree. But I think immediately you can see that the kind of the halwaxids as a for the brachiopod body full plan hypothesis can be more or less rejected on the character basis. We have a grade of Lophotrochozoans that lead up to the major groups of brachiopods that we kind of all know and recognize. And the other interesting result of this is the position of the tomodids as sister to lingulids. But I'll talk a bit more about that soon, because rooting, because rooting, of course, is often a problem in animal phylogenetics, and it's kind of the source of much of the controversy in the within the brachiopods with molecular data in the past having indicated that brachiopods were paraphyletic, with essentially rhynchonelliforms as the root, whereas morphology and more recent molecular data that I'll talk about later producing this topology. So this seemed like kind of a hard problem until this little critter turned up, Eugenetheca elegans, again from the Zhang Lagerstätte. And this is kind of a classic. This was published earlier this year. And this has a combination of both phoronid and brachiopod characters. So I think when this, animals like this are coded into the matrix, so this was kind of an early version that we included in the paper, which had this thing falling outside kind of the brachiopod crown group. But when we actually look at the cladogram that from the entire data set, what we recover is a kind of total group brachiopoda, kind of plus Eugenetheca, which has this kind of combination of odd morphologies, and phoronid's a sister to a monophyletic brachiopoda. With, again, the groups here are just highlighted, the lingulids with essentially an origin in the tomodids, as it seems, and the craniids here, sister to rhynchonelliforms. But again, your mileage may vary with this. It's still early days, version 1.0. And how does this actually compare to broad-based phylogenomic studies? And what is interesting is that there seems to be now kind of a broad congruence between the at least the deep splits that essentially supporting lophophoric monophyly, so that the lophophore seems to have a single origin. And this is a structure shared between brachiopods, pheronids, bryozoans, and that form a clade. This is also supported in a slightly more in-depth version of the brachiopods, this paper from Sperling et al. And it also identified kind of what the issue with the molecular data has been, that essentially fast-evolving sites and heterogeneous evolutionary rates can bias the tree topology. And so with phylogenetic signal dissection, you recover, again, a monophyletic brachiopoda with a phoronid sister group. And just for fun, I actually ran the some of the Cohen et al. Data sets in Raxmal right before I came. It turns out that you can actually recover the kind of the similar consensus with a proper rooting position, but the brachiopodologists will notice among you the issue here is the position of the craniids is now sister to the lingulids, which is in conflict with the morphological data. But we don't live in a perfect world, and the other caveat to note is the bootstrap support in this node is relatively low. So this kind of presents an opportunity for total evidence, because morphology and genes operate on a different scale, as pointed out by Gonzalo Girabay. So I think it's kind of time to start actually combining these data sets and actually think about what the implications of character mapping onto these trees actually means in terms of how brachiopods evolved. So just to kind of provide a general summing up, these discrete characters applied to tomodids actually seem to have kind of a wider distribution than initially thought. We think that we can find some good characters with good homologous information here. The monophyly of Lophophorata is broadly supported by the molecular data, and the morphological data seems to also indicate a monophyletic brachiopoda. So there's kind of broad consensus at the base. And we also have some new exciting fossil discoveries. Lars Holmer will talk about a typical early phoronid as well from the Zhang Lagerstätte. So arguments that phoronids aren't there, therefore the derived brachiopods are now no longer valid. And in terms of the internal relationships, we think tomodids with the initial version are sister to lingulids, essentially. And the answer to other deep questions about Lophotrochozoans may lie in the early fossil record of small Shelly. So it's worth looking at these things. But the caveat to all this is that the character support is quite weak. As a result of this, brachiopods are quite a character-poor group. So as always, more information is needed, and a few possible future scenarios are to look at this in terms of time-calibrated phylogenies, improve taxon sampling, and look at ancestral state reconstructions. But again, the kind of concluding point is that, fossils really don't make sense until we actually code the data, analyze them, and come up with a testable hypothesis of what they are. So this is just a little quote from my colleague Greg Edgecombe. So that pretty much wraps it up. Just thanks to my co-authors and following people.